Voy a comenzar con el anuncio de seguridad. En caso de cualquier emergencia, vamos a evacuar el auditorio en dos mitades. Esta mitad sale por esta puerta y la otra mitad por la otra puerta. Las personas que estén cercanas a las puertas nos ayudan a abrir las dos puertas, ambas hojas, para que podamos evacuar con facilidad. También les pido por favor que mantengan libres los pasillos de cualquier mochila o cosa que nos interese espacialmente. Y a partir de este momento les pido por favor que apaguen sus celulares y los pongan en modo silencio para que no interrumpamos la presentación. El día de hoy tenemos un seminario especial. Va a durar un poquito más de lo acostumbrado, va a ser alrededor de 1 hora y media. Y la presentación la va a hacer el doctor Jorge Nico.
Gracias, Carolina. Welcome to Mexico for our visitors from KAUST. I hope you really have a very productive and wonderful experience here in the country, and in UNAM especially. I'm going to shift to Spanish for your introductions. I'm going to speak with the audience about why you are here, etc. Bueno, pues buenas tardes, gracias por asistir aquí al seminario de los doctores visitantes de KAUST. Les voy a dar una muy breve introducción de por qué están ellos aquí.
Espérate, tú normal.
Muy bien. ¿Se oye?
Sí.
Muy bien. El año pasado tuve la oportunidad de visitar con una pequeña delegación de México KAUST y es impresionante. Es la manera más fácil de definir la universidad. Es muy nueva, tiene creo que apenas va a cumplir 10 años de creada y tiene una planta académica internacional de muy buena, de una altamente alta calidad. Igual el estudiantado es internacional. La idea de esta universidad es pues llevar a Arabia Saudita el máximo de conocimientos de la ciencia y la tecnología ahí mismo, a su propio país. Y bueno, la idea de esta visita es de que ellos quieren ampliar colaboraciones, en particular con la UNAM. De hecho, varias de sus visitas de entre hoy y mañana, todas van a ser dentro de la UNAM, varios institutos. Y la idea es plantear posibles colaboraciones entre laboratorios, pero también el poder captar estudiantes que puedan hacer allá estancias, en algunos casos pueden ser cortas, en otros casos pueden ser ya más enfocadas a hacer un posgrado. Ellos ya les dirán un poco de qué se trata. Entonces, tenemos tres ponentes hoy. Tenemos al doctor Salim Al-Babili, que va a ser nuestro primer conferencista. Después va a seguir la doctora Ikram Bilou y al final tenemos al doctor Rod Winning. Entonces me voy a permitir presentar a cada uno de los tres para ya luego no hacer más introducciones y que podamos fluir más. El doctor Salim Al-Babili se doctoró en biología en la Universidad de Freiburg en Alemania. Después hizo un postdoctorado en la Universidad de Rockefeller, donde fue además tuvo un grupo de investigación, y él es más conocido por haber contribuido a una publicación muy interesante en el campo de la biotecnología que se llama el arroz dorado. Y esto tiene que ver con la producción en el arroz de altas cantidades de carotenos, bueno, que son precursores de la vitamina A, perdón. Él es un excelente bioquímico, biólogo molecular y sobre todo se ha dedicado a estudiar reguladores del crecimiento vegetal como las estribolactonas, y su interés es utilizar estos conocimientos para combatir parásitos de la raíz en plantas. También está interesado en el estudio de reguladores naturales del crecimiento y su señalización en las plantas. Actualmente es el jefe del programa de ciencias vegetales de la Universidad KAUST y es profesor asistente y ha publicado por lo menos 80 publicaciones muy importantes dentro de su campo. Bueno, posteriormente tendremos a la doctora Ikram Bilou. La doctora Bilou estudió en Marruecos, ella es de origen marroquí, hizo su doctorado en España, de hecho habla muy bien español, así que cuiden su idioma, es broma. Ella estudió en Granada, ella estudió en el CSIC, en el Consejo Superior de Administración Científica, un doctorado, y posteriormente hizo postdoctorados en la Universidad de Utrecht, en Holanda, en donde posteriormente consiguió una posición como profesora asistente, después fue asociada y llegó a ser además miembro del Departamento de Biología del Desarrollo y del Grupo de Ciencias Vegetales de la Universidad de Wageningen, también en Holanda. Desde el 2016 se integró a la planta académica de KAUST, en donde ahora es miembro de la División de Ciencias Biológicas y Ambientales y Ingeniería, donde dirige el Laboratorio de Desarrollo y Biología del Desarrollo y Biología Celular de Plantas. Por último, tendremos al doctor Rod Wing, quien apenas acaba de llegar a KAUST, tiene 2 semanas que lo contrataron. El doctor Rod Wing, tuve el placer de conocerlo cuando éramos bastante jóvenes, estábamos de postdocs en la Universidad de California en Berkeley. Él hizo su licenciatura en la Universidad de California en Berkeley en el 80, posteriormente obtuvo el doctorado en la Universidad de California en Davis y hizo un postdoctorado ahí en el Plant Gene Expression Center en los años 80 y parte de inicios del 90, que ahí fue donde coincidimos. Posteriormente se fue como investigador, como asociado de investigación a la Universidad Cornell, y desde el 91 se movió ya como profesor a la Universidad de Texas A&M. Posteriormente estuvo en la Universidad de Arizona como profesor, de hecho fue su más reciente trabajo y aparentemente todavía sigue ahí, está con permiso como para estar en KAUST, y a KAUST llegó hace 2 semanas. El doctor Wing también tiene una gran cantidad de trabajos, sobre todo en genómica de plantas, y entre sus logros hay uno muy interesante. Fue el empleado del mes en 1974 en un McDonald's. Vean qué orígenes tan humildes tiene, y llegó ahora a ser miembro de la American Association para el avance de la ciencia, ¿no? Ha obtenido muchos reconocimientos y bueno, pues sin más los dejo a ellos para que nos platiquen sobre KAUST y sobre sus investigaciones. Al final va a haber preguntas. Sí, las preguntas mejor las dejamos hasta el mero final.
Thank you very much, Jorge, for this nice introduction and for inviting us to this beautiful place. We haven't seen so much by now, but that what we saw is really very impressive. Before I start my scientific talk, I want to show you some slides about KAUST, starting with a short movie about our young university.
Discover a place unexpected, a place where the improbable becomes possible, where conventions are challenged and new ideas are born. Discover a place where your capabilities reach new heights, where your ideas converge with others to make a lasting mark, where there is a shared passion for doing things that matter, to change the world through science and innovation. Come to a place where your curiosity can unleash new discoveries, where ideas launch startups and dreams build new technologies, where competition drives collaboration to make lives better. Discover a place for your passion and potential. Discover KAUST.
Can we start the slides, please? That's KAUST, that is where we live and work. There's a young university built in 2009 by Abdullah. We have a surface of 39 million square meters. That is the community where people are living, where we have schools, hospital, supermarket, and so on. That is the town center. Is not so busy as the town center that we have over here in Mexico. This is our campus where we have the institutes and the labs, etc. And here we have companies and innovation cluster that could actually complement the mission of the university as a scientific institution. These are some numbers about KAUST. We have now by now 1,050 students, 150 faculty, but we are in a growing phase. We aim to have around 250 faculty in the next 5 years, and student numbers of around 2,000. We have 430 postdocs, 300 research scientists, and around 1,500 alumni. We are very diverse. So in the community, we have people from 108 countries, and the workforce from 83 countries. In total, we are having now around 7,000 community members, and a significant number of them are children. There is an excellent place to grow kids. We have around 37% of our students are female. The students are divided into MS students and PhD students. We don't have undergraduate students. And here you see the distribution of the students from different countries. We have around 38% from Saudi Arabia. We have a target of 40 to 50% Saudi students, not more. That is, need to be international just to keep the university as an international university. We have many students from Asia, China, and rest of Asia, from Americas, US, Europe, and the MENA region, Middle East, and North Africa. The university has a mission to work on areas that are important for Saudi Arabia and the whole region. We are working on food security, on increasing water resources, on energy and environment. These are the major targets of our research and teaching. We have 3 academic divisions: the biological and environmental science and engineering, that is our division. And we have the computer, electrical and mathematical science division, and the last one is the physical science and engineering division. Each division has different teaching programs and research center. The research centers complement the work of the faculty by focusing on very applied projects and targets. That is, again, here you see the campus, you see the institutes. I'm going to show you a few slides about our division. We are around 45 faculty. Most of them are in the bioscience program. We have people working in the Red Sea, in the marine program, and then environmental and science program, and in the plant science program. The focus of the division is on understanding how organisms sense and react to environmental changes. This work leads to fundamental research activities that is at understanding the adaptive mechanisms at genomic, molecular, cellular, and signaling level. At the same time, we use this knowledge to develop modifying technologies that aim, for instance, at increasing the food security or the resources of fresh water. At the same time, we try to translate our understanding of biological processes that occur under extreme conditions, for instance, in brines of the Red Sea, translate this into biotechnological approaches, for instance, by identifying enzymes that work under high pressure, the high salinity, and high temperature. Recently, we have developed a program to target the biomedicine that was not very well represented in the past. Here again, we are focusing on the response of organisms to their environmental changes. In this frame, we have a large epigenetic program. Regarding scientific disciplines, our faculty are working in cellular molecular biology, microbiology, and we have biophysics and bioimaging, bioengineering, marine science, plant science, and water science and technology. We are having by now 4 programs. The bioscience program is the largest one, and the marine science program, plant science program, and environmental science and engineering program. But we are establishing a new program about bioengineering that will start next year. The plant science program has the mission to understand how plants grow under harsh conditions and to translate this understanding again into technologies and approaches that can help improving growing crops under harsh conditions. KAUST offer fellowships for students that want to make MS or PhD. This is quite attractive, I would say. The university pays a full tuition. Students get annual stipend. They have free housing, free medical and dental coverage, relocation allowance. They get also a ticket to fly to their home places every year. And if they have children, they get free daycare and free schools for their children. Of course, we want good students, so we have a few selection criteria that runs from good GPA over sufficient, very good English knowledge. In addition to this regular student program, MS and PhD program, we have a visiting student research program where we offer students from around the world to work on certain and defined research projects for 3 to 6 months. Here again, we ask for a certain quality of the students, and we offer many benefits that compensate their work in these defined research projects. Here you see our plant science faculty. We are now 7, working at complementary techniques and approaches. Rodwin will speak later. Also, we are having Magdi Mahfouz is working on genome editing and developing new technologies to improve the ability to edit genes and genomes. We are having Simon Kattinger, he is a serial genetist with focus on pathogenicity in wheat. We are having Mark Tester, a plant physiologist, working on salinity. He is also, in the last few years, has been focusing on developing quinoa to a crop that can be planted around the world, and in establishing new technologies that utilize salt water in greenhouse horticulture. Harry here is a biochemist and microbiologist. He is working on identifying microbes that help white plants growing in the desert to survive under these conditions, to collect these microbes and use them again to increase crop productivity. And he's working on innate immunity in Arabidopsis. Plant biochemist and molecular biologist. Today I'm going to speak about our last work on identifying carotenoid-derived signaling molecules and hormones and translating this knowledge into quite applied project. Plants are effective communicators. They don't speak, but they communicate using chemical signals. They communicate with microbes, with fungi, and also with other plants by sending and sensing chemical signals. Apart from this chemical signal, they use a different type of compound. These are hormones to coordinate all processes of their life from seed to seed. Many of these chemical signals and some of the plant hormones originate from this pathway, from the carotenoid biosynthesis pathway. We know carotenoids are as essential constituent of photosynthesis. as pigment that compare their color to many flowers and fruits. Here you see the diversity of carotenoids that arise from this pathway in stereo configuration, in number of double bonds, and in the presence of the cyclic amide groups. Besides these general functions as intact molecules, carotenoids are targeted by oxidative cleavage, that results from the presence of double bonds. This oxidative cleavage leads to a variety of compounds depending where the cleavage has taken place on the enzyme that decorate the primary cleavage product. So you cleave the double bond, you get one aldehyde, one carbonate, two products with the oxygen function in each of them. You see, you can get something that you have in your aftershave, that is beta-adenine, something that you need to see, that is retina, coming from the same skeleton, which is just determined by the site of the oxidative cleavage. Here you see examples for plant hormones that derive from carotenes. We have strigolactones, and we have the famous abscisic acid that regulates abiotic stress response of plants. The cleavage process is usually catalyzed by a class of enzymes that we call carotenoid deoxygenases. You see here the skeleton of a carotenoid. You see the iron that is the cofactor of this enzyme. Here, oxygen gets access to the substrate. The site of the cleavage is determined of the double bond that is actually exposed to the iron. Here the substrate is entering, here the product is released. We have a hydrophobic patch that allows the uptake of carotenoids from the membrane. Carotenoids are lipophilic pigments. They are usually present in membranes. My original interest in carotenoids was actually generating crops that have increased provitamin A in edible parts, such as the goldenrod that is now almost 20 years old, but finally we got this product admitted. Later on, I started investigating the biosynthesis of hormones. Here you see the functions of strigolactone. These are a novel class of plant hormones. They regulate different aspects of plant architecture. They are mainly known for their role in determining the branching, shoot branching. If a plant is lacking strigolactones, it will develop more branches and show dwarfism in the shoot growth. Our strigolactones were originally identified as signals released by plant roots and abused by seeds of parasitic plants such as striga to germinate and to coordinate the whole germination and growth process with the presence of a host that is required for survival of these obligate parasites. You see the life cycle of striga, sensing of strigolactone, building of the so-called haustorium, then getting in contact with the roots. They would siphon off water and nutrients and products of photosynthesis. They will grow and be happy, and the host would suffer. That is the structure of strigolactones. You see they have a quite complex structure. We have the ABC skeleton coupled to a second lactone that we call D-ring. We have the amino bridge. There are three types of strigolactones. When I start studying the synthesis of this compound, they were known to derive from carotenoids. We knew that there are two carotenoid cleavage enzymes involved in the process, a further polypeptide of unknown function. Then we have a cytochrome 450 that is localized outside the plastis in contrast to the first three enzymes. And we have here the product strigolactones. Chemists would predict here at least 9 steps to produce this skeleton and further steps to produce the other part and to get the final product by coupling both of the two molecules. That is the result of our work on the biosynthesis. We showed that the B27 is an isomerase and that the formation of a compound that we call caractone that is quite similar to strigolactones requires the activity of only two enzymes, these two carotene-cleaving deoxygenases. I'll not go into details here. A very amazing point is that the CCD8 is an unusual enzyme. If you offer an altrans substrate, it would introduce oxygen, produce this product. If you offer a 9-cis configured substrate, you would get this unusual product with 3 oxygen functions. That is the pathway, how it looks today. It is what we call calactone. We show now that calactone is intermediate, and it is the precursor of all types of strigolactons. Besides the known hormones and signaling molecules that arise from strigolactone or from carotenoids, there are many lines of evidence that suggest the presence of further signaling molecules that are synthesized from carotenoids, including unidentified growth regulator. We are trying to identify these compounds by using 2 approaches. The first one is we identify carotenoid modifying and cleaving enzymes. We look for mutants or generate them, study the enzymatic activity, identify the product, and look for its activity. The other one is we test the activity of known and predicted cleavage product and try to find out which of them have bioactivity as regulatory metabolite. That is an example of a compound that we found just by screening for predicted cleavage product. We call it ancarine, that induces the formation of a certain type of arabidopsis root that are called anchor roots. And that is a second signaling molecule that we call saxinone, which we identified by studying the enzymatic activity of a carotenoid cleaving oxygenase that we call saxinone synthase. You see the corresponding mutant is really growth retarded, has also more telomeres and less panicles. Here you see an example how we can detect the enzymatic activity just by detecting the change in color that is caused by cleaving and shortening the conjugated double-bond system. We further investigated the activity of saxinone and found out that is a negative regulator of strigolactone biosynthesis at transcript level. It down-regulates the activity, the transcript level of the first, second, third, and fourth enzyme in the pathway. This leads to a less release of strigolactones. Here you see the mutant, the wild type. If you just count the number of strigolactones that arise when you plant the mutant and the wild type, you can actually just predict that the mutant is releasing much more strigolactones and that this compound is a negative regulator of strigolactone release. Now to the application. Striga is a severe agricultural problem, particularly in sub-Saharan Africa. It affects cereals. Estimated losses go from $7 to $10 billion a year in maize, millet, sorghum, and also rice yields. And it's actually it's one of the 7 major biotic threats to global food security. A major issue if you want to control striga are the release seeds. One single plant can produce up to 200,000 seeds. These are spores actually rather than seeds that survive for more than 1 decade in the soil. You can imagine that infested soils have accumulated a huge number of seeds over the years. You see just the comparison between the seeds of striga and the seeds of the host. Here you have millet, sorghum, rice, and maize. One possibility to deal with the seeds problem is the so-called suicidal germination. That was a hypothetical strategy that has been discussed for decades and applied probably only 1 or 2 times. You see here in the here above what happens usually in normal infestation cycles. Plant would release strigolactones, germination of striga, then attachment to a host. One could exploit this dependency on the host by applying strigolactone analogs. In the absence of a host, this would lead to germination of the seeds that would die because of the absence of a host. We call it suicidal germination. If we want to use this strategy and test it in Saharan Africa, we need actually efficient and affordable strigolactone analogs, something that people can buy and you can apply to soil and then we see effect. And we need an application protocol for rainfed agriculture because we don't have so much water to just spread the compound around. When we discovered calactone, we found out that it is actually biologically active. This was surprising because it was predicted that the compound must have this immune way to have activity as a strigolactone analog. Therefore, we decided to use this calactone skeleton to produce strigolactone analog that can be applied to combat striga in Africa, and we got support from Mr. Gates for this story, and developed calactone-based analogs. Here you see different structures that we tested as analogs. These are similar to calactone derivative that we call calactonate. We tested these compounds in the lab by determining the number of striga seeds that germinate, and we get quite promising results with 2 or 3 of these compounds. Here you see the receptor binding activity of the strigolactone analogs. And then we went to field. We tested this compound in Burkina Faso at 3 different locations. And we developed a protocol where we rely on rain to distribute our compound in the soil. This means we wait for the first or second rain, then we apply our compound. This will be further diluted by the coming rain that will bring the compound to the seeds that will then germinate and die. And here you see the result that we obtained. Here above in the permanent millet field, here below in the sorghum field, we have significant reduction in both cases, which actually depends on the nature of the compound. What was active in the sorghum field was less active in the millet field. And we are focusing in our project on permanent. Permanent is a very important crop for arid and hot regions. This is the 6th globally important stable crop, and it is actually of a very high nutritional value if you compare it with rice or other cereals. And it's grown by people who actually cannot grow anything else, and of course it's highly affected by striga. We are investigating 2 contrasting lines. We found out that the susceptibility of this cultivated line is actually depending on strigolactone pattern. This was also clear by investigating, so it's dangerous. By investigating the architecture of both types, we are sequencing the genome of both. We have just got funding for a 5-year project from the Gates Foundation. Here we want to further develop our strategy of suicidal germination by developing new strigolactone analogs. We want to develop compounds based on zaxinone that accelerate the growth of plants and downregulate the release of strigolactones. And we want to sequence the variety that we have, the contrasted two lines, and identify genes and factors that contribute to striga susceptibility. Thank you very much.
Thank you, Salim. And our next speaker is Dr. Ikram Bilu.
Hola a todos, buenas tardes. Ante todo quiero agradecer a Roberto y a Andrea la ayuda que nos han estado dando esta mañana y agradecerlo a todo estar aquí, a ese país maravilloso. Ayer estábamos en el museo y habíamos aprendido muchas cosas. Vosotros tenéis una cultura muy buena, hacéis mucho esfuerzo para mantenerla, que es muy admirable. Now I'm going to switch to English, and the talk today will be about. Can you hear me? If I ever speak loud, because I like to move while I'm giving the talk. So what I would like to. So what I would like to talk to you about is the root system architecture. So the idea is to how to go from pattern research, from basic research, into understanding adaptation into desert conditions. So for a long time, I have been studying mainly focused on. So I was mainly focused on the basic research to understand how a root become a root. So why do you have in a plant? Why do you have a system which is roots, which is responsible for acquiring nutrients and water, and not becoming a shoot which is responsible for photosynthesis. Let's see if this is. Okay, so the focus of the lab, so we have many projects running at the moment. The first one is the networks regulating asymmetric subdivision and protein movement in the root. And what we can see from here is that we have a root system here with different proteins that are expressed in different cell types. And one special feature of the plant, so that the plants cannot move and they have the cells that are stick to one another because of the presence of the cell wall. And because of that, so cells have to communicate with one another, and they created a mode of communication which is intercellular movement. And one of the features that is special for some proteins is that some proteins move from one cell to another, and during this movement what they do, they convey positional information. So they carry information from one cell to another so that the cell know what to become in the future. So what we are doing at the moment, understanding how these proteins are moving, what are the mechanisms that are restricting this protein to only one cell layer, so why don't the protein move further, and how does it impact growth and the formation of different cell types. And so we do this in roots, we do this in shoots, and then in lateral roots. Then we have the other project is mapping protein complexes in vivo. So proteins are interacting with one another, and these interactions are really important for many biological functions, including transcriptional regulation, including cell fate specification. And what we are doing for the first time, we managed to create a protein map in which we can see how two proteins are interacting with one another in living organisms at the cellular resolution. And with this finding, what we managed to do is to find specific complexes that are acting in different cell types, and depending on the conformation of a complex or pairs, what we will have is different genes will be activated, and that's what we will give the cell fate of different cells. Then the other project is how a plant decides when to defend and when to grow. So for instance, when it's attacked by pathogens, plant have to choose. And this choice is either basically not much choice. So usually when it's attacked, all the resources are allocated to defense. And what we want to understand is how to uncouple these two processes. So how can we create plants that can grow and at the same time defend themselves? And this we are managing by dissecting the role of the bird proteins, which are transcription factors, which we found that they have a really important role in mediating both growth and defense. And for the last is, since we are now in the desert, so one iconic plant is the desert plant, which is the date palm. And what we are trying to understand how these plants in the deserts have acquired the ability to adapt in desert conditions using the tools that we have learned from fundamental science. We are now using them to understand growth mechanism and adaptation to desert conditions. So plants, the plants have need to grow, and to grow they need food. Of course, they need to acquire nutrients, they need to acquire water. And to do that, so they need an efficient root system. And this is a very nice illustration of how complex is a root network in soil. So this is an image of a tomato root grown in a rhizotron, and where you can see different networks, nodes, and these network nodes are digitally made, and they represent how every laterals in the roots is acting as a source for uptake for water and nutrient uptake. So to get this final root system architecture, what you need is you need a healthy root, and you need to have a healthy root like this. This is rice and this is Arabidopsis. What you need is a proper tissue type. You need a plant that divide and differentiate in a timely manner and in an optimal manner. And to know or to understand how this complex system is made. So it's really important to understand how does it come about, how does it start, what kind of the mechanisms that are involved in maintaining growth. And to understand how the growth is maintained, you need to go from the basic and to see what kind of cells are made, are produced, and how are they specified. And using Arabidopsis as a model system, what we have been working in the past year is to understand how different cell types are specified. And what you can see here, for example, we have in Arabidopsis, we have the stem cell niche, and the stem cell niche is really important because it's the place where the cells are dividing and differentiating to give rise to different tissue types. For instance, if you have here a cell that will divide, and then it will give rise to the epidermis. And we know that the epidermis is the one which is having the root hairs, and the root hairs are really important for uptake of nutrients or minerals. Then we have the vascular tissues, and we know that having a proper vascular tissue is really important because that's where everything is getting transported. Then we have the endodermis, and the endodermis is the place or is the tissue type where there is a kind of a hydrophobic barrier, which we call the Kasparian strip, which prevents many pathogens from entering and also prevents water from escaping from outside the roots. So understanding this basic mechanism of how a root is made will help us to at least make sure that we have an optimal root growth, and that will promote or will help us to have a better understanding of how to make plant grow better and stronger. So here I will give you two examples. So these are how the roots change in response to the environment. And here is a root in which, so this is a maize root from Cosidinelli's lab. And so what was done here is that they put the root in soil, and they made a kind of space where the roots do not have enough nutrients, because then there is a hole in between. And then, on the contrary, if these roots are growing normally, what you can see that you have a really optimal root system. So to say, you see the differences between these two plants. So if you don't have a system which can acquire optimally your nutrients, so the plants will not develop optimally, and then you will not get proper growth. So it's really important then to have a proper, an optimal root system to promote growth in general. So now I will move to a more different topic, which is understanding the adaptation strategies that date palm are performing in desert conditions. And then why date palm? Because date palm is very important for many cultures, especially the Arab culture. It's a very ancient plant. It dates from 4000 BC. It can make kind of thing that we call desert farming. It creates kind of hierarchical farms. So you have here the palms, which create some shadow, and then you will have the farmer will create, will be able to plant his fruits and vegetables. So you can find here wheat, you can find many tomato, etc. It's a plant that can survive very harsh conditions, and of course the fruits are really delicious. And in many cultures, for example, you can find it in the Bible. It's the tree where Maria was eating the fruits while giving birth to Jesus, etc., etc. So I would like to start with this movie, where we started to look at the root system architecture by looking X-ray micro city. And what you can see here, that first the roots develop normally and start to make kind of anchor roots. And with this technique, what we managed to have to notice that there are some structures that usually are not present in normal plants, like for example, roots that will go at the direction which is opposite of the gravity. Usually we know that the root will grow downward. And these structures that are called pneumatophores, which are usually used in plants like mangrove to provide aeration, and then and in the case of date palm, we think it's a strategy to maximize water uptakes when there are rainfalls which are really rare. Then in this, okay, the movie doesn't work, doesn't matter. And here in this movie, what you will see is that the plants, during the first phase of germination, you have things that is like roots, and then it will germinate, and then the shoot appears much later. And that's something specific for the palm, because they germinate in a mode that is called remote germination, which is germination at distance. So first the seed will set something that is called cotyledonary petal, which is this one. And then after a while, it will go downward, reach the gravity, and then reach the soil. And afterwards, a series of succession of developmental events will occur. For example, here you have a seed that germinates, it will go, and then it will carry the baby plants within the soil. So you can see here at an early stage of development where you have less divisions. As you can see, with the green is marking the cell division, the nuclei that are in S phase, and then after a while, the cell division will start, and then they will be still in this protective layer, which you can see here, in this layer surrounding the whole plant, carrying the whole system in soil. So, unlike all the plants where organogenesis usually occurs above ground, in date palm organogenesis occurs downward ground, and this is most likely another way to protect itself from attacks from predators, from environmental conditions. And what you can see here at this stage, you have the meristematic genes, the genes that are involved, that are specifying the root and the shoot and the boundary expressed at the same time at this stage. And interestingly, this mode of germination is not specific to it's specific to date palm, because if you look at the oil palm, which is a closest relative, it has completely different way of germination. You have the shoot and the roots completely separated. And we know that, as Salim has just said, plants are, there are many hormones that are involved in promoting root growth, and the interaction of plants with environment, like for example, the microbiome is really important for promoting the root system architecture, and also for protecting it against things like salinity and drought. And in a close collaboration with Daniele da Fontio in KAUST, what we did is we looked at the effect of bacteria that are isolated from desert palm, and what we found is that these bacteria could enhance the root system architecture. So if you look here at the roots without endophytes, you can see that the structure is well developed, but it increased when it's in the presence of the bacteria. And so this is a root of Arabidopsis showing that the bacteria can colonize it, so it might have an effect. And interestingly, when we treated this plant with salts, what we found that unlike outside or non-treated control, in presence of salt you have a limited growth. This, in presence of the bacteria, the growth was not impaired, so suggesting that this endophyte might have a beneficial effect on enhancing the tolerance of plants in certain conditions. So then what we ask, how does it work? What kind of mechanism is involved in this process? So it was something very easy to test. We know that auxin is involved in promoting root branching, and that's what we have been seeing. So we just looked at the control and the bacteria, and this is a root with DIR5, which is an auxin marker, so it's marked, the green color marks the auxin distribution or the growth regulator auxin. And in the presence of the bacteria, what you can see is that the auxins become highly expressed, so you have an increase of auxin distribution in root treated with bacteria. And of course, once you see this and being very familiar with the pin protein, so we looked at how the bacteria is affecting, because this is really clearly saying that you have a defect in auxin transport. So we looked at the pin proteins, and what we found is that the pins, instead of being localized only in the membrane, in the presence of the bacteria, we have the pins becoming in these vesicles, which are which colocalize with the endocytic marker FM464. And so what does it tell us? That the pins usually that are recycling from the plasma membrane into the vesicles, when treated with the bacteria, the pin proteins recycle faster. So if they recycle faster, then they would be auxin would be transported faster and then accumulated much higher in different tissue. So to have a sample model, we have the pins in normal conditions, and for example, pin 2 is apical and basal in the epidermis and the cortex. In the presence of the bacteria, there will be a substance that is produced. We think that it might be auxin, but we are not sure which substance it is, so maybe Salim will help us identify this molecule. So once this molecule is present, we have pins that are completely mislocalized from their positions, and then and leading to an increase in auxin transport. When you have increase of auxin transport, you have more lateral roots, and when you have more lateral roots, you have more tolerance to many developmental stresses. So we started now developing tools to monitor plant growth in a non-invasive manner, so that means in a natural condition. And this is using just a normal camera, so what we call it mobile imaging, where using different light spectrum, what we can do is look at a macro scale level at things that are really in contact with the roots, so that will allow us to see what kind of changes the roots in the roots are occurring under different conditions. The condition can be anything, so you can go to this level. So this is a worm in which it's eating, and then you can go much, and this is not just a normal camera, so we're not talking here about a microscope or a very cheap. So you can go and then look at the nematodes that are really invading the plant. And with these tools, what we are doing now is trying to understand how plants are responding to different parasitic interactions like nematodes or even the parasitic weed striker. Okay, I will skip that, and I would like to acknowledge the people that have contributed to the work. For example, like Tintin for the date file. Then our collaborators across the world, and also our collaborators in KAUST. And of course, the group members, so this is the last desert trip that we have. So, so this is our team. So this team working on date palm, beans in plant-parasitic interaction. Then the plant pathogen is taken care by these ladies. And of course, all this data would not be here without the help of all our collaborators, either in the state or especially our colleagues in KAUST. And then the last thing is, if you are interested to join us, you will enjoy this view. For example, you can do snorkeling, you can do diving, you can also see beautiful things in the desert, like looking at the desert, the camels, and then you can go to the Maldives without going to the Maldives because this is in Saudi Arabia.
Thanks, Ikram. Now it's turn of Prof.
Okay. Yes. Hi everybody. It's wonderful to be here. I just moved to KAUST in February, so it took me to go to Saudi Arabia to come to Mexico City. So, all right. Let me see if I can figure out how to write this thing.
Bottom blue.
Sorry. Okay, that's that. Okay. Oops. I just spilled it. Sorry. I'm actually giving you my talk, but I want to start from the beginning. How do I start at the beginning? Okay. Okay, sorry about that. Our lab is interested in the big question is essentially how do we solve the 10 billion people question, and our group is working on trying to harness 15 million years of evolutionary history from the genus Rhiza. Okay, so our big question is, again, how do we grow enough food to feed the world by in less than 40 years? Rice is going to be a major component of that. Rice feeds half the world already. It's a rice-dependent population that will double by 2050. So we're working on the development of new varieties of rice that are higher yielding and more nutritious, and that have less of an environmental footprint, ones that require less water, fertilizer, and pesticides, and have reduced greenhouse gas emissions. We call these green super rice. Okay, I'll get it. So this is an outline here. I'll talk a little bit about natural variation, domestication history of African rice, 13 genome data package, and some future directions. So the way that we're trying to attack this problem is by trying to understand natural variation, right? And in rice you can find diversity in rice for almost any conceivable trait. This shows photographs of seed color, seed size, seed shape, panicle architecture, plant architecture. So I have a joint appointment at the International Rice Research Institute in the Philippines, and they have a gene bank that is composed of 130,000 accessions that have been collected from around the world. And recently we were involved in a project to sequence 3,000 of these accessions. When we did that, one of the big conclusions that came out of that work was that those 3,000 accessions, when that data is mapped to the reference genome of rice, you can now subdivide that those 3,000 accessions into essentially 15 subpopulations, or kind of, I like to call this like 15 races. We have the Japonica, Indica, the KAUST types, and then those are subdivided even further. So one of the things we're interested in doing with that data is now trying to associate that data with phenotype. And Matthew Morell at Erie has proposed this idea called the global rice array, where we take these 3,000 accessions, plant them around the world, and under different environmental conditions, and do baseline phenotyping, and then collaborate with people around the world to do topic-specific annotations, like annotations with respect to root disease, flowering time, yield, so on and so forth. So this is a field in Erie. We're already doing this. We're doing phenotyping using tractors and drones, capturing traits like canopy cover, height, days of flowering, biomass, so on and so forth. What we're interested in doing now is importing that material into Arizona and putting this in what's called the field scanalizer. It's in Maricopa, it's near Phoenix. This is a gantry system right here where you can, this has wheat right now growing under it, but it goes back and forth this way, and then you have the sensors going here. So Arizona has a lot of land like this, and we're going to be using a machine like this as well as phenotyping on the ground. Similarly, we're going to try to be doing something like this in Saudi Arabia, and possibly maybe even having a collaboration with folks in Mexico to try to also establish a joint center for desert ag. Another form of natural variation that we're looking at comes from the wild relatives of rice. The wild relatives of rice represent 15 million years of evolutionary history right here. We have 27 species. We have a wide distribution in habitat. We can find rice in Asia, Southeast Asia, Australia, the wild relatives in Africa, as well as Central and South America. We have 11 distinct genome types. We have the AA genomes here, which is the primary gene pool for cultivated rice. And then we go all the way down to the GG genome here. We also have 3.6 genome size variation. So the rice, the AA genomes are around 400 megs, and we can go all the way up to about 1.8 gigs. So what I like to say is that the wild relatives are essentially agronomically inferior, but contain a virtually untapped reservoir of genes that could be used for crop improvement. Here is Asian rice, African rice, and these are the wild relatives. This is Rufipogon here, thought to be the progenitor of Asian rice. One species I like to talk about a lot is Rhiza cortata. It's a very distant relative. It actually is a halophyte. It can grow in salt water. Has a range from Myanmar to Pakistan. And scientists at Erie have actually successfully crossed cortata with Asian rice and have brought in some of the salt-living pathway. What we're interested in doing there is sequencing that genome, which we've done, and looking at the molecular genetics of salt tolerance and transfer those genes into cultivated rice, as well as try to domesticate Rhiza cortata. So to understand the wild relatives of rice, I initiated a project back in 2003 called the Rhizomap Alignment Project. That's morphed into the International Rhizomap Alignment Project, as well as the Rhiza Genome Evolution Projects. And one of our long-term goal is to create a genome-level closed experimental system to study the genus Rhiza in both basic and applied aspects. So these are our collaborators here from around the world, including France and China and Australia and the States and Brazil. But a majority of these folks were involved in the original rice genome sequence back in the early 2000s. So IOMAP, we've kind of had several meetings around the world, and we've had three basic things that we're interested in. One is generating high-quality genomes and transcriptomes of all of the species of the genus that would be representative, developing mapping populations for functional breeding studies, and identifying collections of natural and recurring accessions for diversity, population, and evolutionary analyses. So one of the first genomes that has come out of that is we published this in 2014, and that is the African rice genome. So the reason why I like to show this slide is that this is called Black Rice by Judith Carney, a professor at UCLA. This is a story about slavery in the United States, and there was a time in the 1700s where South Carolina was the largest exporter of rice in the world. And the question is, really, how could that be? And the slave owners would say they taught the slaves how to grow rice, but in reality, the slaves came from West Africa in regions that had been growing rice for over 3,000 years. So her book is essentially about that slavery was not just about muscle, but it was about the transfer of knowledge from one culture to another. So we try to tell that story in our genome. And one of the questions that we asked was, did early African farmers select for the same genes that early Asian farmers did 7,000 years earlier? So Asian rice was domesticated in Asia about 10,000 years ago, African rice about 3,000 years ago. So we did a series of tests, and we identified 5 genes that appeared to be associated with domestication in Asian rice that may have been selected for an African rice. The DEP1 genes involved in panicle architecture. These genes involved, RC genes involved in seed color, semidorph in plant height. The BAD1 genes involved in aroma, and shattering genes. This is a photograph of wild panicles. When they are the seeds mature, you shake the panicle, the rice goes everywhere. Cultivated rice no longer shatters, so it's easier to harvest the rice. So what we did was we looked at 2 shattering genes in rice. One was SH1. This shows the configuration of the genes in the shattering locus in Oryza sativa. This is the shattering locus here. We have 5 upstream genes, 2 downstream genes, and there's a single point mutation that conveys non-shattering in Oryza sativa. We looked at Oryza barthi. This is the progenitor of African rice, and they're separated by about 500,000 years. They have the same architecture here. We look at Oryza glaberrima, and essentially there's a 40 kilobase deletion that knocks out the shattering gene as well as 3 additional genes. So what we concluded from this experiment is that, yes, indeed, early farmers selected for the same genes, but the mutations were completely different. So this would be kind of considered convergent evolution. So I'll skip this second gene. So since that time, we've developed, we've sequenced 13 more genomes, and we call that the 13 genome data package. These are the genomes we've sequenced here. We also, that was on the IOMAT team, primarily my group. We developed, annotated for transposable elements, developed transcriptome data sets, baseline transcriptome data sets, methylation data sets, and then we did a common annotation across the whole piece, and then did topic-specific annotations on NRL genes, new genes, and structural variations. So I'm not going to really go into that. I'll just say that this was published this year in February in 2012. But the reason why I wanted to show you this photograph was that I'm going completely the wrong directions now, going backwards, showing you the whole story. After when our paper was accepted into Nature Genetics, I submitted this drawing from my daughter, who drew this as a t-shirt for the wild relatives of rice. That was their dance team. And I said, this is never going to get accepted because of this naked rice right here. but in reality it was published. So I always like to thank my daughter for doing that. Katie, when she's 16, so we co-published together. All of our data can be found at green.org. And then in terms of future directions, one thing we're interested in doing is, sorry guys. One thing we're interested in doing is establishing a joint center for desert agriculture between KAUST and the University of Arizona. And then last night I was thinking, well, maybe we could do something similar with UNAM. And that would be pretty exciting to do something like that. So that there might be a way that we could have a three-way situation where we're phenotyping the same sets of, it can be rice, but it can be other crops like maize as well. And I think this is pretty advanced in maize, but doing this as a desert agriculture collaboration would be pretty interesting. The other thing we're interested in doing is, again, sequencing the remaining species of rice, especially the polyploids here. We're going to be doing that this year, so by the end of the year we'll have full genome sequences for all representatives of all 27 species, and we have a lot of work to do on that. And then cortada, again, we want to collect voucher specimens along its species range to do population analysis, and we want to try to domesticate the Rhizia cortada. And one thing I wanted to go back, I think I'm going to go back one slide here. Another thing that we're really interested in is I'm interested in establishing a collaboration with scientists in Central and South America to collect some of the endemic species to this region. There's 4 species that are in this region, so that might be an opportunity to, as well, to have an interaction with scientists here in Mexico. So with that, I just thank a large team, our funding, NSF, AXA, Tanimar National, NIH, and again, this is how I see the world. And so I recently moved to KAUST, and again, we're recruiting group leaders, postdocs, and graduate students. And I just wanted to say that if any of you are interested in any of our programs that we talked about today, or any of the other plant science, or just coming to KAUST to check it out, Lucy in the audience right here has an iPad that you can give her email, and she'll we can contact you, because there's some real good opportunities. So I came to KAUST because there's a lot I want to do, I still want to do, and KAUST is going to give me the long-term funding to make that happen. And that's a big deal, because I've been writing grants for forever, up and down and up and down, and always trying to figure out where the next dollar is. Here you can get long-term support to do the projects you want, and we want to have the best graduate students work with us to make this happen. So with that, we'll leave it open for questions for everybody. Thank you very much. Maybe we were to stand in the front. Anybody's interested?
But we have time for some questions.
We can say no. Any questions? No questions. Anybody interested in coming to KAUST? We got a couple of people filling things out.
Pueden preguntar en español y traducimos. Si tienen cualquier pregunta.
Bueno, primero agradecerles que hayan venido a Seminarios y que es una muy buena aportación el hecho de que se interesen en colaborar con esta universidad. Y bueno, la pregunta va enfocada a cómo este proyecto podría ayudar en general a combatir la deficiencia alimentaria en el mundo y particularmente en África.
need some help with that.
Yeah.
Who's it to?
Yeah, so first she was thanking for your presentation, but she's thinking that you're coming all the way here to UNAM. And the question was, how do you think that these projects can help tackling the lack of food in general in the world?
How
How can your projects can help tackling the lack of food?
So again, with our, at least on my side and on all of our side, we're essentially just trying to, learn about plant biology, learn about development, learn about the biochemistry of plants, and also learn about natural variation. And we're trying to combine all that to create new crops that are, again, higher yielding and more nutritious, but have less of an environmental footprint. That's really kind of what we want to do, and that's what I'm deeply interested in. And I'm trying to capture this by, I mean, I think GMOs have a role, but it's a lot of times it's just like one gene at a time. Whereas over 15 million years of evolutionary history, there's been trial and error for that amount of time. So we already have so many different versions of genes and genes that have not yet been discovered yet. And we want to discover those by studying this Erie gene bank and then applying that to make combinations and unique combinations to make new varieties of rice that are, we call it green super rice. So that's the direction we're trying to go. And you can maybe add.
From our project, we have a very targeted problem. We are developing solutions for it from different sides. We have the biochemistry on one side, and we have the genetic part.
But this striga is a huge problem in agriculture, and solving that will be.
I mean, it's one of the major ways to keep security.
Well, first of all, congratulations, very interesting talks. And as a mycologist, I was wondering if you have any experience or have you thought of incorporating marine fungi to the research on salt tolerance?
Marine fungi?
Yeah, marine fungi. For example, maybe marine endophytes.
Marine endophytes? Why marine endophytes? I ask you.
Well, because they already have the adaptive traits to cope with salinity, perhaps.
So that's, with respect to like the rice cortata,
Yeah.
for sure, that's something that we're interested in looking at that microbiome as well, and trying to see if we can have a combination of the two. For sure, but I mean, that would be interesting to isolate the endophytes from cortata, for example, and try them on land plants.
It doesn't necessarily to be a result of the microbiome or endophytes. We have many examples for plant and
The problem with marine endophytes is how you need to have them grown in the niche environment. And these endophytes are probably grown in the sea, deep in the sea, so to bring them up into desert or is going to be very challenging.
Well, we have some experience with, for example, with mangrove or seagrass endophytes. So, as far as I know, as far as I've tested in my lab, it's possible to isolate those fungi and to keep them in culture.
Oh, yeah. So we have a huge mangrove area all around KAUST. And so I've been running around there. I've been looking and said maybe we should try to sequence some of these genomes and look at some of their endophytes as well. So maybe that could be another collaboration for sure. Absolutely.
We have one more question here.
Hi, this is a political question. I was looking at your first talk and you mentioned a bunch of different patents. And I am wondering who keeps the money and the funding from the patents? Does KAUST keeps it? Does UNAM keep it if you go 50-50? who keeps the money that comes from those patents of all the research that you're patenting?
This was your talk. I think when you introduced about the, no, I'm sorry, you didn't talk. It was your talk.
Yes, you mentioned a bunch of patents that you
Patents
found out.
belong to the university, and they are going to license it if you can develop it to a product.
Yes. So if you have a collaboration with UNAM, how does that split?
That will be split.
Would be still 50-50?
Yes.
80-20? How does that work?
Depends on the project and the input. You know, how much each partner has put input in the story. So for instance, I have a patent with a colleague from Japan. So KAUST has 75%, University of Tokyo has 25%. In other cases, we have 20% in KAUST and 80% in the partner. So it depends on the input.
And that input is financial input, or is it biodiversity input, or how do you calculate it?
It's intellectual input.
Intellectual input?
Yes. Definitely. And that's going to be negotiated in a collaboration contract.
But I think Mark Tester was saying that the patent rights are pretty generous in KAUST, right? I mean, quite a bit of it goes to the investigator. It doesn't. I think some universities, most of it goes to the university, but they get KAUST. It might be like 50% goes to the investigators. I don't know the exact number, but it's pretty generous.
I did translate my patents yet.
Okay. But we're not lawyers, so.
But they are humans. And they want to see a product, so they always looking for the license.
Okay, thank you. I think we are up to the time, so we'll have some time for discussing further during the coffee, which will be in this side. Thank you very much.
Para aquellos estudiantes que estén interesados